Hi guys. So we are going to talk about cellular respiration today, and I think you're really going to enjoy this a lot. So we're going to review a little bit about what chromosomes are, the eukaryotic cell and cycle, and mitosis in general. And then we're going to leave meiosis and variation to chromosome structure till later. So let's talk about what the chromosome is. Now, the field of genetics is anything that involves dealing with DNA, right, which we talked about last week. Cytogenetics is specifically looking at the chromosomes within the cell and cell division specifically. So when we get ready to actually divide to make another cell, you actually see the chromosomes become compacted. So if you remember when we were originally looking at cells in roughly about week 4, we were looking at the nucleus and we saw it stored as chromatin, which is that kind of squiggly lines. Now, when we want to go and divide that nucleus in half and divide the cell in half, when we want to make new cells, if they get old or they get worn out, something like that, then we need to basically compact that DNA so that it can move easier. And you can actually see these by light microscopes. Now, if we want to look at those chromosomes and we want to look at number or the size or the form of the chromosomes, then that's what's known as karyotyping. Now, humans have 23 pairs of chromosomes. So we get one set from our moms and one set from our dads. And for each of those 23 chromosomes that we get from each parent, for instance, chromosome 1 that you get from your mom has the same genes on them as chromosome 1 from your dad does. So they pair together. So essentially you have a total of 46 chromosomes, right? The first 22 are what are known as autosomes, and those are going to be the ones that don't have anything to do with sex, right? The last pair is our sex chromosomes, and that's what's going to make us either a girl or a boy, okay? And so when we're looking at cells or the number or sets of chromosomes we have are called ploidy. And so a diploid cell, do you see how it says di for 2, right? And ploid. So this is saying that essentially we have N is the number of chromosomes, right? Here. And so by itself, right, we have one set, and so 2N here is what's considered a normal cell, right? And what I mean by normal cell, I mean a somatic cell. And that's anything like liver cell or skin cell or anything like that. Haploid is denoted by N. And this is just one set, right? So this is going to be something like a sex cell. And can anybody give me an example of a sex cell? It would be sperm or eggs. So diploid, 2N, somatic cell, right? Anything in the body besides sex cells. And haploid, which is just N, right, are sperm or egg. And so that's how we're arranged chromosomally for humans. Now, when we're talking about homologs are just talking about, that's when we were talking about the two sets, that every chromosome from your mom has one that you pair up from your dad, and that's what's called a homolog. So think homo as in same. So essentially, those are what are known as homologous chromosomes. So all your autosomes, which is, what does autosome mean again? It's the first 22 chromosomes, right? Each of those are homologs of each other because one comes from mom and one comes from, you're right, dad. Right? Sex chromosomes aren't essentially identical in genetic composition, which essentially we're looking at, right? The autosomes, if you have, for instance, on a chromosome, if you have a gene that color is eye color, then you're going to have the genes for on eye color for mom on a chromosome, and then you have the homolog with gene color from dad, right? Where sex chromosomes, essentially, they look a little different. The X's for the girl are going to look different, but the X and the Y here, the Y will look a little bit different than the X does. The Y is actually smaller. Now, this is what we call a karyotype right here. And this karyotype, do you see here? These are the homologs. Essentially, this is chromosome 1, 2, 3, 4, 5, 6, 7. And as you see you going down, they get smaller, right? And so you have one here that comes from mom and one that comes from dad. You can see they're about the same length and size. And they essentially are going to have the same overall genes. Now, whether the genes can look differently, as in you might have a gene for brown eyes on one and gene for blue eyes on the next. So it doesn't mean that they're exactly the same characteristics on each gene, but they're the same gene. And basically how we do this is they can take it from blood cells, basically centrifuge it down to make this like pellet here. We resuspend that in a hypotonic, which makes the cells burst, if you remember, and then you can put and stain them on a blood slide, or I'm sorry, not a blood slide, on a slide itself, right? And then you can use a microscope, and now they have technology. You can see it, it's on this computer screen right here, to be able to look at them. If you look here, though, you'll notice they're not lined up. A computer program actually lines them up really nice. They used to have to do it the hard way, where the scientists would look and like cut them out and pair them up. Now there's computer programs that do all of that for us. But this is a perfect, if you get an amniocentesis, if you're pregnant and over the age of 35, 36, you're considered a geriatric pregnancy. They'll want to look at your chromosomal counts and look to see if there's any abnormalities. So this is where you'll see something like a karyotype being done, and this is what they're doing to look to see if your child has something like Down syndrome. So if you remember what chromosomes looked like here, remember they started out as chromatin. So chromatin are pieces of DNA and protein come together, and this is what ends up making chromosomes, right? This is, if you remembered inside the nucleus, you saw all the different lines. And if you remember, this is what chromosomes look like when they're normal, not dividing, right? Now, chromosomes are going to be allowed for us to sort DNA, i.e., go into cell division, right? And so they go into daughter cells, right? They'll be parent cells, and they'll divide out, and essentially they will become offspring or daughter cells. Now, they can either be duplicated when the chromosomes essentially start sorting or condensing, and that means they're just going to be a linear strand, which means they literally just look like this. If they become duplicated, then essentially they'll divide and there'll be a new one. Like on one side, there'll be an old one, and then the new side, it'll be a new one, right? And genes are what's actually on this piece of chromosome or DNA, and these are informational units that tell us how to make proteins. And so this is an organization. So you start essentially with this long piece of DNA, and normally it's all over the place, right, in the DNA. And this is the double-stranded, and you can see it goes around. And these little circles here are what are known as nucleosomes. And the proteins that hold them around that circle are what are known as histones. And so they can pack tightly. Do you see how they're going zigzag back and forth? And so they'll basically come really, really close to each other. And then from here, there's these things called scaffolding proteins that help hold these in place while these kind of move and kind of whirl back and forth. So it makes these looped domains or these pieces of chromatin. And then essentially that chromatin, do you see here, kind of moves all around. Well, this area that's in here gets smaller when it becomes condensed. And then that essentially, this is like a cut up area of this right here, getting really, really close. And so it's going to end up being a condensed chromosome later. So that's how we actually make the chromosomes that you're used to seeing. So nucleosomes are basically these circles, right? It's the same idea. Okay, guys, if your grandmother or anybody you know likes to do like any kind of knitting, they'll have yarn. And the yarn is everywhere. It takes up a lot of space. So what they'll do is they'll wrap it and condense it into a ball. And then it makes it very easy to transport, and it keeps it from tangling and being just a big mess. This is kind of what's happening here. And so the histone proteins are the proteins that help to basically allow the DNA to wrap around, and it kind of coils, right? And then it's held together by scaffolding proteins. So this is a close-up of this C double-stranded DNA, and then basically it goes a couple times around and goes to the next one. So this whole thing here is what's known as a nucleosome. These little purple things are the histone proteins that keep it. This is what's known as a linker DNA, and that's just the DNA between two nucleosomes. And that helps to basically keep the DNA structured. So this is what all the little nucleosomes look like. You can see these little circles here, right? And it looks really, really clean here, doesn't it? And that's because these things are whirling around, and there's all that scaffolding proteins there holding all of that in place. Well, let's remove the scaffolding proteins and see what happens. Oh, Lord. Do you see all of this mess over here? Do you see all of this? Everywhere? Look, you can barely even look in it. You can see these are the little lines of DNA right here, and how much there's kind of, how much there is compared to, look at that. And then let's look back at this. Do you see how different that is? So the scaffolding proteins help to make it less of a mess in here. And just to give you some perspective on here, in each cell, there is 6 feet of DNA. So we have to try to, put PT on there. We have to try to figure out how, or the body needs to figure out the best way to get that DNA in there without causing a mess. This looks like if you have any kind of holiday lights that you didn't put up correctly, then you get them out and they're all tangled. There's no way that we would be able to use this efficiently to make a new strand of DNA quickly. So it would get all knotted and tangled up, and it would be a mess. So that's where nucleosomes, those histone proteins, and the scaffolding proteins help. So this is what a chromosome looks like here, right? And so this is a duplicated chromosome. So normally you would see here one side, over here, and over here, right? You'd see one side, but we've actually duplicated this. So each side is what's known as a sister chromatid. If you notice, we were talking about chromatids earlier, now it's tids. And I like to think this is when we actually divide. And the only way we're going to be able to divide is if we've D for duplicated. So this is a duplicated one. So this is a sister chromatid, that's a sister chromatid. Now what it holds the two in place is the centromere, or here, right? And then when we go to move these around to divide, it's going to be kinetochores here, or this outside region. that basically the spindle fiber microtubules are going to come out so that we can move these around. So, and I think it looks like kinetics, which is the movement. When we talk about energy or movement, kinetic energy, and kinetics has to do with movement. So hopefully that helps you there a little bit. Now, prokaryotic cells, all this that we've talked about with eukaryotic cells so far is slightly different in prokaryotics. And this is the only time I'm going to mention prokaryotic cells. Essentially, they have what's called binary fission. And this is a way, it's a lot like mitosis, which we're going to talk about here in a minute. The biggest thing here, essentially, is they're going to have DNA, and it's just going to replicate. And this is what's known as a plasmid. They're circular pieces of DNA. And we're going to talk more about this when we get into biotech, okay? So essentially what we're doing here is making a new piece of DNA just using this process called binary fission. So let's talk about what's considered the cell cycle. And we probably have about 5 or 6 minutes left of this, and we're going to be done for the night. So the cell cycle is exactly what it means. Cycle as in we're going to go around and start over again. Cell is what we're using. And basically there's this idea that every cell originates from another cell. And that's what's known as a cell cycle. So when we want to grow and get bigger, or we want to replace old cells, we do that by dividing those cells, right? And we know this is extended back for nearly about 4 billion years, and you're going to talk more about this information when you get to Bio 2 and evolution. It's very highly regulated, and there's 2 divisions. There's mitosis, which is for somatic cells, which remember these are things like liver cells or skin cells, and meiosis, which is for sex cells, right? So the one we're focusing on here is going to be for mitosis. So the cell cycle has 2 main phases. It has what's known as interphase and the M phase. Interphase is the prep phase. This is essentially like, if we're moving, this is like the packing and the cleaning of moving. This is the actual move itself. Which one do you normally see from the outside? This one, right? You can't normally see when people are packing and cleaning inside their house. That's why we focus on this one, because this is the one you can actually see. Now, interphase is the longest, right? Think if you're packing and cleaning versus actually the move, which one takes longer? Getting ready for the move. So there's more time spent on this, but this is what we actually see. So this interphase gets divided into 3 parts, G1, S, and G2. Each one of these is going to basically either we're getting bigger, we're making enough organelles, we're making enough proteins, and we're making more DNA. Here is when we start moving those things around into each cell so that we have enough of them. Now there's this phase called G0, right? And that's this 0 right there. If you notice the G's up here, it's basically G0, like almost like it's before this, right? Because this substitutes for G1 if cells are not dividing, right? So G0, I think G not dividing again, right? Because they're done. So let's talk about the cell cycle and what's happening there. So this is what the cell cycle looks like, right? You have the beginning. This is interphase here, right? You can see it. You have G1. This is a period of growth before DNA starts to replicate, right? So here we're basically making more organelles. We're making proteins, getting ready for the next part. We're getting bigger. All of this is happening in here. Now the S phase, think synthesis. This is where DNA replication happens. So you know what we talked about last week with all the enzymes? That's here. In G2, this is the prep stage that is basically your last check. It's making sure we have everything we need before we start the M phase or mitosis. The M phase is basically combined of mitosis and cytokinesis. Mitosis is the nuclear division, where we actually divide the nucleus. Cytokinesis is the cytoplasm division. And you can see PMAT, PMAT. There's actually one phase we're going to add in there called Prometaphase. But that is essentially the cell cycle. So that basically showed you the different phases of mitosis. So let's look just the preparatory phase. Now, interphase is not part of mitosis, right? It's the pre-stage. So here you can see DNA still there in lines. It's not condensed, and it's divided into those three parts. G1, which is gap phase 1. That's where cell growth and prepares for the S phase, right? So we're making new organelles, getting bigger, all of that. The S phase here is where you actually get DNA replication, right? It's called synthesis phase, and that's what happened from last week. And the gap phase is increased protein synthesis so we can get ready for everything, and we're getting ready to divide. So they're going to do a lot of checks and balances here. So the first, the M phase, like I said, is part of mitosis. That's the nuclear division, right? So we're going to end up getting two nuclei or two nucleuses that look the same as the mom and dad nucleus or the parent nucleus. Cytokinesis is when we actually get the cytoplasmic division, and this is when we actually have the final new cell. And this is at the end and overlaps mitosis. So the best thing that I can tell you to help you remember is please pass me another taco. And basically this is prophase, prometaphase, metaphase, anaphase, and telophase, right? And these are the 5 different stages of mitosis. So prophase is the very first, and this is when you're going to start seeing cells dividing here. So the chromosomes condense. The nucleolus in the center, where you normally see that little dark circle, that disintegrates along with the nuclear envelope, right? That starts to break apart. These mitotic spindles from the centrioles will start forming and coming out, and then the centrioles all start moving to both sides. Prometaphase is when your chromosomes begin to actually start moving to the middle, but they haven't gotten there yet. And that's showing you an idea of kind of what it looks like. They're not there in the middle yet. Now, how these chromosomes move is through mitotic spindles. So essentially, remember how this is the kinetochore area? So it's a chore to move. So to move and the chore, kinetochore, right? And so these centrioles will be on opposite sides. And if you see these astral microtubules, they kind of help to hold these guys in place. Now you have the polar. Do you notice they're not attached to anything? The polar are going to keep these pushed at opposite ends. And the kinetochore microtubules are the ones that are actually connected that move the DNA. So that's how DNA is moving. They're actually being pushed around by these mitotic spindle fibers that are basically called microtubules. Now, once they meet the middle, think M, middle, or midline, right? And here are the chromosomes are the most condensed. So this should make sense. Since they're the most finely packed, it's going to be easier to split them or pull them apart without breaking them. Pull apart, without causing DNA damage. Hopefully that makes sense. So that's what's going to happen here. Anaphase, this is where, if you noticed earlier, you saw the chromosomes here, right? This is a sister chromatid, this is a sister chromatid. So at this point, they are going to be pulled away from each other, and the sister chromatids are both going to go to opposite sides of the pole. Telophase is when you actually get the opposite prophase. So the nuclear envelope reforms, the nucleolus appears, the chromosomes uncoil and go back to chromatin. The mitotic spindle fibers start breaking apart because we don't need to move them. And this, you actually get cytokinesis to begin, or starts to begin. Now, the initial forms of cytokinesis can be seen in anaphase, but it gets revved up in telophase. And this shows animal cytokinesis. Now, these guys have contractile rings that basically pinch. It almost works like, what do you call it? Like sweatpants, where you have drawstring. And so it keeps tightening until we cut here. Now this indention it creates is called a cleavage furrow. And this can happen because there's no cell wall for animal cells, so this is easy to happen. Now the problem in plants is they have a cell wall. So we have what's called a cell plate forms. And essentially you get these vesicles that have parts of that have cell wall on them and plasma membrane. And they just start forming these big cavities in the middle until you get two separate cells. So when we look at this, what kind of cell is this? What do you see forming here? This is an animal cell, and we have the cleavage furrow forming here, right? Where here it's a plant cell, and what's forming here? It's the cell plate. So let's look at these different parts of mitosis and see if you can recognize them. So hopefully that helps you. Now the cell cycle, the amount of time spent at each stage does vary between species, cell type, and the growth conditions. But if you're looking at a cell cycle period of 12 hours, G1 would last about 5, S would last about 4.5, and G2 would be 2, right? So we're looking at about 11.5 hours of interphase, which is not part of mitosis. The part where we actually see move would be about 30 minutes. So the prep stage is much, much longer than the actual movement stage. But the movement stage is so important because that also leads to a lot of issues and genetic disorders later that we're going to talk about. And throughout the sequence, there are checkpoints just to make sure that we have all the proteins and enzymes available. For instance, how do we know to get to the midpoint before we divide? And there are checkpoints that do that. And so when we're talking about asexual reproduction or offsprings that are basically single parents that don't combine with anything, they use a procedure much like mitosis to help to allow a new daughter cell to come off a parent cell. And that's what's known as binary fission, and that's what prokaryotes do, like I mentioned earlier. And so this is where we're going to stop. I hope you enjoyed it, and I hope you like what we're going to do in class for activities this week. So I'll see you later, guys.